21. So at 20 itself, he's got the, because at the earlier age, he got the fellow. It's been recognized for his work on the abiotic stress tolerance. Apart from the US, also a DBT Ramalingam Swami re-entry fellowship, because very difficult to get Ramalingam Swami re-entry fellowship. He's also one of the fellow of re-entry fellowship. Actually, Government of India introduced this fellowship because to bring the talents from outside the country. In that, he is one of the successful fellow, and he been here as a faculty in India serving. And also he is having the DBT Innovative Young Biotechnology Award and the Ramanujam Fellowship also he been got. And he have very good publication. That means from his citation 6,142 H index of 38 is a very high. The youngsters has to follow his things, how he is working on that. And also he's guiding masters, PhD and postdoctoral fellows also. So he have a lot of DBT sponsor projects, and he have his lab is working on mainly the origins, many of the defense mechanism, non-host resistance plants against pathogens. And he earlier is working on Arabidopsis model system, Arabidopsis thaliana. Now he's those techniques is to introduce on the plant system. So the audience are wider audience, biotechnology students, and also the breeding and pathology students are there. So they are. I think your lecture will definitely give us some idea on how these kind of interactions really help us to look in a newer look on our plant system. So I'm really happy that he readily accepted our invitation and come over here. So on behalf of Tamil Nadu Agriculture University, I also thank our Vice Chancellor for permitting us to conduct this kind of invited lectures from the experts from all over India and outside the country. So in that, I'm really happy to be here, and let's hear his talk on this area of host plant resistance and abiotic stress tolerance. Thank you. So I welcome
Okay, good afternoon, one and all. I profusely thank Professor Senthil and Professor Kokila Devi for inviting me here, and for the nice introduction, Professor Senthil, and all the august audience here. So without going further into the details, I'll straight get into the talk. Excuse me for the movement. I think I will be able to manage stuff, but maybe I will be a little bit slow. But I'll finish stuff in about 45 minutes. Maybe if there are any questions, then we can take it up after that. Okay, so today, as Professor Senthil introduced, that I'm going to talk about one of the basic plant biology work. It's going to be a bit deep, so I request all of you to follow. It's not going to be heterogeneous. I'm going to talk about only one gene, and I'm going to talk about its interactions with others in that perspective, and especially with the sugar as a source for pathogen nutrition from that perspective. And you can feel free to interrupt me anytime to make sure that if you are not following it up, make sure that you interrupt me and then ask me questions right away. And of course, we can take up the questions at the end if the chair permits further on. Okay, so I belong to an institute called National Institute of Plant Genome Research, which is basically a DBT institute. Most of us get confused with NBPGR, which is an ICR institute, but this is a DBT institute. And we are located in New Delhi, in JNU campus, Jawaharlal Nehru University campus, right next to NII, ICGB, and few other institutions like IIT Delhi. So that's the area we are located in case you are knowing. Since Professor Senthil has introduced about the PhD program, I just want to add on few things like, yes, we invite very good quality students for PhD and afterwards for postdoctoral programs as well. The procedure involves, first you have to have either CSIR or DBT fellowship secured, then you are eligible to apply. So now we have introduced a system of written examination by ourselves. Then after that, in the same day, the results will be announced. then there will be at least a couple of days of interview, then the results will be announced for the shortlisting. Normally, the intake range these days is around 25 to 30, which varies every year depending on the requirement we have. And we offer the thesis problems ranging from bioinformatics, plant molecular biology, genomics, plant microbe interactions, other plant stress biology, plant developmental and signaling. So these are the areas on which that we offer PhD programs. It depends on the expertise of the group. We are around 31 research groups led by 31 different PIs. And depending on their requirement, the research programs are scheduled. Okay, so let me get into the talk directly. You know that the nutrients present in the plant cell or plant is the heaviest demanded one, be it a human population or microbes or insects. So the reason for which that we all land onto the plant is to get nutrition out of it. Today I'm going to talk about why a particular microbe, which we call it as pathogen. For nature, it is just a microbe, but for us, it's pathogen because it damages our crop. Why that microbe sits onto that plant and how it extracts, especially taking an example of bacterial system, prokaryotic system. So I'm going to talk about Pseudomonas syringae, a bacterial pathogen in this perspective. So the Philosphere is one of the good nutrition source, which is emitted out of stomatal excretions, and of course water also comes out of it. Many Pseudomonas lives on Philosphere. Apoplast, this is the main discussion point for today's talk. You know that apoplast is the intervening cellular area that between cells, the region that belongs to the cells where the exchange of nutrition happens, and most of the microbes depend on the nutrition that are available in this area. And I'll show you later, the bacteria completely depends on, it can live only in the apoplast. So I'll show you that. And the second one, the third one is phloem, and then xylem, and also some of the cell enclosing organelles, including cytoplasm, right? These are the places where nutrition is present. Now, I will just introduce one small slide because we have taken a pathogen which belongs to non-host, which is a non-host pathogen, because for the suitability of this study, to get a better explanation of that study results, we have taken a non-host pathogen. What is non-host pathogen, I would like to say? For example, if you say the pathogen 1 here, and pathogen 1, if it lands on a plant 1, it causes the disease. Right. The same pathogen, if it lands onto another closely related plant, it cannot cause disease. Instead, the plant induces a mechanism called hypersensitive response, which is a defense response. It could be possible the vice versa, that the pathogen 2, which is a host to plant 2, can become a non-host for plant 1. So this is what I meant by non-host. When I mean non-host pathogen, the pathogen is currently not able to infect that plant, while it is successfully able to infect its closely related plant. So whenever I mention in the talk about non-host pathogen, this is what I mean by. And when I mean by non-host plant, this is what I mean. So the important reason why we quenched into this topic is that you know that there is a society called American Phytopathological Society, in combination with about 10 societies of this similar regions, came with a series of meetings over a 5 years to 6 years duration. They have debated and then identified what are the top 10 unanswered questions in molecular plant microbe interaction field. One of the questions that they have identified is an answer to what is non-host resistance. So that is where the field of ours is gaining importance, and this is the field in which the important field in which that we are working currently. So I'll just brief. In this case, the Arabidopsis system. Throughout the talk, I'm going to talk about only Arabidopsis. All I'm explaining is going to be only on Arabidopsis. So it can have two kinds of resistance. One is type 1 non-host resistance. Another one is type 2 non-host resistance. The type 1 non-host resistance is nothing but it produces the non-HR type of resistance, which does not produce any visible symptoms. Type 2 is the one that I showed you in the previous slide, where it shows up the hypersensitive response, which kills the pathogen at the site of infection. Of course, this is the typical disease which we call it as host pathogen, wherein the host is becoming susceptible. So the other two are resistant reaction, and the susceptible one is basically a reaction that leads to disease. Now, to just give a glimpse of this, these are the labeled bacteria that are present in the cell taken using a confocal microscope. So we have labeled the green fluorescent protein expressing bacteria, and this we introduced into the Arabidopsis or any other plant. You can take it; it's the same situation. So you can see that the cellular region, the intracellular region that is being focused here is the only region that on which the bacteria is able to be present. Because you know that the bacteria never enters into the cell. It can lyse the cell and it can completely open it, that becomes necrotrophic, but then no more cell exists. So whenever there is a cell intact, the bacteria has to reside only in the apoplast, and it has to take up the nutrition only by inserting stylus, which can again put the factors into it and then make the plant to excrete the nutrition out into the apoplast, then it eats it. So that's why this study is unique in a way that it's easy to distinguish between whether the bacteria is eating or whether the plant is supplying or not supplying. So which you will realize in the upcoming slides. I'll just play one small video of how it's a real-time video of how this bacterial movement happens in the apoplast and how it multiplies and it moves from one kind of apoplastic region is a continuum. So it starts from mesophyll to all the way until the endodermis related part until that. So you can go if it is a root, and if it is a leaf, it goes through all the mesophyll and all other layers also it goes in. So it's a continuum. So the bacteria also has, you know, that it can move. So this is how it moves into the cell. So the main point I would like to say is that you can see the clear shape of the cell, and only in the apoplast the bacteria is present. Now, to introduce this, we see that a host pathogen, which you can see clearly the apoplast is filled with the host pathogen cases that you will not be able to see that multiplication, only the stays inside. Now, in the control apoplast, wherein there was no pathogen was infected, we have taken that apoplast and then could put it in put the bacteria into it, the tube, and allow it to grow. So in the minimal medium, it will be this much. When there is an apoplast that is exclusively supported from a non-infected plant, it grows little more than that, which means that this apoplast has already a restricted availability of nutrition. Now you see in this graph, whenever you put a host pathogen into the plant, right, and then take the apoplast out of it and put a bacteria into it, then you can clearly see that this host pathogen infected plants had a lot of nutrition into the apoplast. That's why it is able to support such a high growth of pathogen. However, you see the main point that I would like to try here is whenever you take an apoplast from a non-host pathogen inoculated plant, you see that it is almost supporting only to the level of a minimal medium that it can supply, which means that this non-host pathogen inoculated plants has prepared well not to limit the nutrition to the apoplast, which means that secure the nutrition in a place where it is not available to the pathogen. This is the strategy that this non-host plants, one of the strategies that non-host plants adopts to limit the pathogen growth. And once you not supply nutrition to the pathogen, the subsequent machinery that are present in the cell, ROS related mechanism and other mechanisms are general antimicrobial compounds can easily take over that defense and then kill whole pathogens. So this is the beauty of restricting the nutrient, making the pathogen weak and then kill it very quickly. So that is one of the strategy that these pathogens infiltrated plants can use it. That's what we are going to study it now. Now, one of the first indications that we got that such a limitation of nutrition can really make the plant resistant to pathogens, we got it from this study. So we were involved in silencing about 5,000 plus genes independently using a technology called virus-induced gene silencing, which most of you might have heard about. And each of these plants were inoculated with the host and non-host pathogens. So in this case, this is the host pathogen, and these are the three non-host pathogens. If you see the non-silenced plant, only the host pathogen is able to grow, and all other non-host pathogens are not able to infect this plant because there is a defense mechanism. So this defense mechanism was offered by a gene called SQS, which is a squalene synthase, which is involved in membrane stability maintenance characteristics. Means when this gene is expressing, the membrane is kept intact and no nutrition is much leaked into the apoplast. When you disrupt this gene, it's not able to produce squalene anymore, and it's thereafter that the other sterols that comes out of it is not able to be produced. The membrane is disrupted. Once the membrane is disrupted, you see that almost all the 3 non-host pathogens that we tested is able to grow, which were otherwise not able to grow, which means that the concept what we were thinking in terms of whether a limitation of nutrition can really result in inhibiting the pathogen growth and thereby bringing the resistance is possible. So now the gene that what we have taken up earlier was a very generic gene. It will just disrupt the membrane permeability characteristics, and it makes the most of the nutrients available to it. But today's talk, I'm going to deal with very specific transporter, where it transports the sucrose and how manipulation of sucrose in the same case, similar manner, that how it will limit and what is the regulation that it happens. That's what I'm going to talk about. So this is just to introduce the concept. When you selectively able to limit the nutrient availability at the site of infection, in this case for the bacterial pathogens, it is apoplast, mainly the mesophil apoplast, it is able to restrict the pathogen growth, means it's able to offer resistance. Now, so this is what the same thing that I have explained it in the previous slides. Now you can see in the leaf anatomy, you can clearly see that the apoplast is all over here. And this is where the phloem and xylem cells happens. And when there is a pathogen entry into the cell, initially it happens in this region, and then later on it multiplies in the apoplast. And then if it is a necrotrophic pathogen, it kills the whole cell and the tissue macerates, and then it takes away the nutrients later on during the necrotrophic stage. So now if you take individual cells apart and see the scenario that is offered against a host pathogen versus non-host pathogen, what this host pathogen is known to do is it's known to induce a style called type 3 secretion system. This introduces into the cytosol transiently, and through it supplies some of the effector compounds. So these effectors are now able to manipulate a transporter called SWEET, which is well studied in rice. It's been well known in rice that these transporters, I think some of you here are working on rice SWEET transporters. And that's why this selective transporters, this pathogen is able to induce it and then make the transporter functional to make the sugar available at the site of its infection. See how clever it is that it's making sure that the cellular sugar that is available inside is leaked out, not leaked, it's in this case systematically pumped out, like using a pump. Like this is the sugar transporter pump, and it now has a lot of the sugars inside of its select sites. And also we know that now the pathogens are known to selectively induce the transporter that is responsible for its sugar of preference. So this is what the system that is going on with the host pathogen. Let's come to the non-host pathogen. In this case, instead of pathogen, the plant is doing the resistance job. So what it is doing, whenever there is a non-host pathogen is attacking the plant, the plant now knows that, okay, there is a pathogen clearly standing outside. So what it does, it now wants to deprive this pathogen out of nutrition. So it just downregulates a particular SWEET transporter, which the plant knows that this pathogen is needing that sugar, and then makes the sugar less available in the apoplast, and hence the opposite strategy, it is able to utilize it and make the pathogen starved of the nutrition that it really needs it. Now the story is now starting. The whole work was beautifully done by one of my first PhD student, who is now abroad doing postdoc, Uruj Fatima. The entire set of data that I'm going to show today was single-handedly generated by this student. So I always show her picture first before I start my actual presentation. So as you know that this system is Arabidopsis, and the pathogen that we are using is Pseudomonas syringae. Okay. Now I'm going to talk about SWEET transporters, as you know by now, that in Arabidopsis there are 17 SWEET members available, 17 different SWEET the Arabidopsis encodes. And it broadly can be divided into hexoses, the transporters that transport hexoses, sucrose, and other different kind of hexoses from the clade 4 category. Now, without further introduction, I will just tell generally the SWEETs are responsible for what are the functions: seed development, pollen development, glucose availability in the vacuole, because this is one of the storage place where normally the cell uses to store it and retrieve it later, phloem loading, and pollen development, freezing tolerance, fructose. So many other factors also I'm trying to say that these SWEET transporters are normally involved, apart from the one that I'm today talking about, that is pathogen resistance. Now, what we have done to start with, which is the sugar transporter, we should start working on it to prove the mechanism that whether the sugar starvation can be a real mechanism. So why I am insisting on the sugar starvation as a mechanism is because you know that in conventional immune responses of the plant, PTI, ETI, or gene-mediated resistance that are important thereafter are conventionally known ones. So far, all our research is focused on that, and a lot of research publications are coming out of that. But I'm trying to propose a parallel kind of a defense mechanism by restricting the sugar availability in the plant. That could be a parallel sugar metabolism, which could be mediated by one of the mechanisms that previously known. But this mechanism of sugar deprivation is the least explored part. So what we have done, we have got the mutants of all the 17 sweet genes and then tested against one of the bean pathogen, which is a non-host to Arabidopsis. It infects the pulses, but it doesn't infect the Arabidopsis. And you can see that in most of the cases, the differential response is clearly seen. But the red arrow marks clearly shows that AT sweet 11, which shows the mutant shows much lesser multiplication of the bacteria. And the other mutants, namely sweet 11 and sweet 12 and sweet 15, there it shows more bacteria, which means that the absence or the knockout of this sweet 12 and sweet 15 has more sugar, in this case, what we can assume it, and hence it facilitated the multiplication of more bacteria. And you see that the 11 is doing, the mutation of 11 is doing the opposite. For now, let's only think about this much. Let's prove the remaining part also, and then we can come for the discussion. So what next we have done is that when we got 3 or more of these sugar transporters that are showing differential, whose differential regulation are playing with the immunity, now we wanted to test individually to dissect which is the most important one or which combination has to be further worked upon to dissect the mechanism. So you can see that Colombia is a wild type plant, and we have developed 8 sweet 11, 12 double mutant, 11, 15 double mutant, 12, 15 double mutant, and 11, 12, and 15 triple mutant. Now you can see the response. Whenever you see the same pathogen I'm talking, the non-host pathogen that otherwise whenever you infect double mutant of 12 and 15, you can clearly see that the non-host resistance is compromised. Whenever the 12 is silenced and along with 15 is silenced, this compromise is much more. However, you can clearly see whenever this 11 is mutated, whether it is along with the 12 or it is along with 11 and along with the 12 and 15, whenever the 11 is mutated, it's not able to offer more nutrition to the pathogen. So you can clearly see that the mutation of 11 is always opposite, less prominent compared to that of either 12 alone or 15 alone, or for that matter 13 also. Now, yeah. So you can clearly see that the mutation of 12 has produced even to the extent of disease symptoms, which means it shows the extreme form of susceptibility. Now, when we did this assay, the first thing what we have to prove is that whenever we show a knockout phenotype, we also have to make sure that when you put back that gene into its native status, whether in that mutants it replaces back to the wild type level, right? That's what first we have to show. So then what we have done, we have complemented this 11 gene in the 11-12 mutant, 12 gene in the 11-12 mutant, and then we also kept the single mutants as it is. So you can clearly see that through this assay, whenever there is a complementation is done, this C11, that is the transporter 11 that is complemented in 11-12 double mutant line, was almost equal to that of 12 alone, which is basically the susceptible response. But whenever we complemented 12 with the 11-12 mutants, it was very much similar to that of a single 11 mutants. What basically it is telling is that when the 12 silencing is leading to susceptibility, if you put back 12, then it is bringing back to the wild type, which is kind of a resistance. However, when you silence the 12, when you silence the 11, which is otherwise when silencing shows the resistance, when you put it back, it's almost equal to that of a wild type, which is expected. Now, the next step, what we have done was that we have to know actually where is this protein localized, and where is this trend of response in terms of its protein expression, the quantity of protein expression, as well as its localization, how it varies. So the first thing what we did was that we have taken a native promoter of 11, that is sweet 11, and fused with AT-suite 11 Gus. So you have a Gus fused with 11 protein that is expressed and driven by the native promoter expressing native promoter of 11 expressing, and then similarly the native promoter of the 12 that is driving the fusion protein of 12 and Gus. Now let's look at the scenario. This is a mark situation where there is no pathogen involved, and the middle panel is the host pathogen which produces the disease, and the last panel, that bottommost panel, is the non-host pathogen which does not produces the disease. So now you can see at the different time levels like 0 hours, 36 hours and 48 hours. You can see that this 11, that is the 11 transporters, is normally well expressed without pathogen, right? Similarly, whenever the host pathogen is exposed at 0 time points, of course, you see the same trend. You can clearly see that as the host pathogen infects, this level of 11 expression goes down. The protein is not localized and its level is going down. And if you put the non-host pathogen, almost you see the similar response. Now, the interesting thing comes with respect to 12. So 12 is very less expressed under a control condition without any infection. But the moment you put with the host pathogen, you can see the consistent expression of this fusion protein of 12. And with the non-host pathogen, the important thing to be observed is that initially it pops up, but later on its levels are going down. So this is the trend that we are seeing. So now we can try to correlate that what is the expression pattern of 11 and 12. So you know now that the 11 is a normally well expressed one, and it is also induced to some even by the pathogen. Later on goes down due to the later stages of infection. But with respect to the 12, you can clearly see that under the normal conditions, its expression is very less, or almost negligible at this case. And whenever you put a pathogen, its expression goes up. And it changes its patch distinction whenever the later stages of non-host comes. You can clearly see that going down after some time. Now, the next thing important we have to prove is that, and by the way, that the previous ones, you should note that this expression is only selectively located in the phloem region, right? So you can always see that the major and minor veins are the only things that are highlighted with the blue color, which means that its expression is kind of restricted to phloem and xylem. To some extent, only the phloem. I wouldn't say xylem because this is much lower. So this again means that this localization is specific. This 11 and 12 is getting localized to the phloem predominantly. Now, the next step, what we have done is that if it is restricting to a particular cell type, that is phloem, whether in the subcellular region also, whether there is any selective targeting of this available. So within a cell, what we have done is that we have taken the labeled plants to know whether the pathogen exposure triggers whether it's targeting to a particular organ. For example, in this case, we are showing that it is targeting to the plasma membrane, which is again to prove that these 7 transmembrane transporters are all having unique features that are related to only particularly, for example, the plasma membrane. So that proves that to prove that. You can clearly see that in the mark conditions as expected. I'm showing only for the 12 data. Of course, level 11 data will be different. So under the normal conditions, it's very less expressed, and you can see that the localization is going to the plasma membrane. But the moment you put the PSP, that is a non-host pathogen, you can clearly see that after 48 hours, its targeting is much clear. And of course, it also happens with the host pathogen. So this, again, just to prove a point that it is localized to phloem cells, and within the subcellular region, it is localized to plasma membrane, which is expected because these are all 7 transmembrane membrane-bound proteins, and whose expression is always targeted to membranes to facilitate the transporter function. Now, the sucrose levels in the apoplast are the key factors. One should quantify it to prove whatever we have shown previously in terms of molecular biology, right? To make sure that whatever we are showing it is indeed translated into the sucrose levels. For example, if I claim that that particular transporter is localized, targeted to the plasma membrane and targeted to the specific cell tissues of the phloem type, and then it is performing the function of sucrose transport and making the pathogen more available with the nutrient. If I have to know that, then the first thing I have to do is that I have to really quantify and show that indeed the sucrose levels are different, and the way that I'm explaining is actually fitting into the sucrose level quantification. So that's what we have done. First thing what we have done is, since we do not know exactly, although these transporters are kind of predicted to transport the sucrose, it may not necessarily just do only this. So initially we have done a complete metabolome sucrose profiling, and then we narrow down to the sucrose. And I'm showing the data for the sucrose here. So you can clearly see that under the control conditions, in the mutants of 11, and wherever there is a mutation in the 11, the sucrose levels are low. So this is expected that in all the 11 mutants, you saw a resistant phenotype, which means that the sucrose levels were less, and hence the pathogen was not able to multiply much. And even in the sweet mutants, even without pathogen also, you see normally a good level of sucrose present. More interestingly, whenever you put a non-host pathogen, the spike is much higher here. You can see that the sweet mutants only had sucrose. All others even had a negative level of sucrose, which means that this is a relative comparison. It's not actually negative. It's a relative comparison compared to even lesser than that of a wild type plant or even a control plant. So which again clearly proves our concept that this PSB multiplication that I showed you in the previous slides is due to the presence of higher levels of sucrose. So now we can say, based on the apoplastic sugar in the control and in the infected in the infection conditions and the pathogen multiplication data, we can clearly say that whenever there was a 11 mutant plants were observed, the sucrose levels were less. And the apoplastic sugar after infection did not change, but you know that the 11 did not have a sucrose in the first place. So even if without infection, if it doesn't change, it doesn't matter much. So you clearly see a decreased pathogen multiplication. It is fitting into our hypothesis of sugar deprivation. The sugar sweet 12 mutant, again, there was no change in the apoplastic content and you in the control condition, but you can clearly see a higher level of sucrose that is spiked up here, and it was consistent with the increased level of pathogen multiplication. Now, in the double mutant, this is where the interesting thing comes. See, this is showing more growth of pathogen, and the 11 is showing less or even much lesser than the wild type thing. So whenever there is 11, it is a kind of a dominant. You see, whenever you mutate 11, you won't even allow the 12 effect to be seen. So it's highly decreased, which is actually surprising, which is much lesser than even the wild type plants. So 11 is a key determinant, which is actually playing the role in controlling the probably the sucrose levels in the apoplast. So now the next thing what we have what we are trying to show is that by putting back the sucrose levels, whether we are able to get the situation back. So just to prove that, what first thing we have done is that, okay, by the way, before I get into that, these are all the ones that are multiple evidences were added to the same the previous experimental results because of a lot of reviewer comments, because they now wanted to know that indeed whether on real time your sucrose depletion, which is happened by absence of transporter, and its pathogen multiplication, can you show it lively on on a cell, in a live cell? So what we have done was that we have developed this marker assay is available, but for this we customized it for this pathosystem, Arabidopsis and Pseudomonas system. So that wherein we can clearly show that this pathogens, so this is the host pathogen that is illuminated, the fluorescence clearly shows that, by the way, the fluorescence is actually highlighting the availability of sucrose. So what we have done here is that there is a agrobacterium called Agrobacterium rhizogenesis, and which is having a marker which can sense the sucrose. So whenever you put that agrobacterium into a media or a tube, and if you supply with sucrose, it will eliminate more. And if you deplete the sucrose, it will eliminate less. In other words, we can clearly say, as a biosensor, we'll be able to show or measure the quantity of relative quantity of the sucrose in a system. So that's what we have applied it in a live apoplastic space. So we introduced the sensor expressing Agrobacterium rhizogens. And this sensor expressing rhizogens will now show the levels of sucrose as indicated here. And we also have, that the green GFP labeled bacteria. So now we'll be able to correlate between these levels in the same cell. Both are measured in the same cell, same tissue. So we'll be able to now show that on a real-time basis, whenever there is more elimination that is indicative of the sucrose, we are also able to levels of bacterial growth also more. So this is actually a proof of the previous ones, which in a in vivo system we are able to show. It took a long time for us to establish this assay and then get this done, but it wonderfully worked out well. And which again even strengthened our own belief that such deprivation of sucrose even whether it is considered as sweet transporters involvement or not, we are able to control by regulating the sucrose levels. We are able to control the pathogen multiplication as well as eventually the occurrence of disease symptoms as well. Now, the next question we have to answer to further strengthen this concept is that they have asked us to put back the sucrose into the place and then show us that whether it can able to retrieve it back to the wild type level. So that's what exactly we have done. So we have taken two different levels of sucrose concentrations, that is 0.1% and 1%. So it's biologically higher concentration if you look at per se, but the amount that once it gets in, how it gets reflected is still reasonably biologically relevant. Now we have taken the non-host pathogen, this is PSP, and then did the same in-planta bacterial multiplication to show how much quantity of the bacteria is applied and then multiplied. And then we also have the plants whose phenotype that produces whether susceptible or resistant phenotype. Now we have a lot of treatments here, 11 mutation, 12 mutation, 11 plus 12 mutation, and the two different concentration levels. To sum up this whole thing, since you know the story already, I will just sum it up in one go, that whenever the AT-SWEL 11 mutant, whose phenotype was generally resistant, if you put back the sucrose, it lost its resistance. It becomes susceptible. And the sweet 11 and 12, which was also having a higher level resistance, when you put back the sucrose, its resistance is lost. And of course, if you put it into a 12 mutant, which was already susceptible, the growth was much higher. So this is how we concluded that there is a direct correlation between the transporter, the apoplastic space, sucrose, and the bacterial multiplication. So these can be easily connectable now that to explain a point. Now, the next thing what we have done was that actually they wanted us to show indeed whether your transporter is able to really having a ligand binding ability, and indeed can you show that the sucrose is transported through your transporters. For example, in this case, we have taken 11 and 12, of which we have given prominence to 12 simply because we suspect that this 11 is only a transporter that is doing a job of somebody's command, most likely by the 12 and whose mediation through some other signaling that is from the top, that is coming from the membrane initiated signaling. So this is just to show that how a deduced structure of this 11 and 12 is located, and we just mapped the sucrose moiety, and we were able to show that the cavity is able to bind the sucrose molecule, and we are able to show this is actually a separate paper. I'm not going to detail much on this. We are able to show that yes, this transporter is actually having the cavity that the ligand can, the sucrose can bind and then come out of after forming the structural orientation of the transporter. Now, the next thing what we have to prove is to biochemically to show whether these transporters in a heterologous systems like yeast is able to do the same kind of expected job of this transporter function in the system. So in this case, of course, we are not going for pathogen or anything like that. We are just coming into a regular biochemistry that what are these functions of these 11 and 12 transporters. Now, the reason why we came into this is that you might have seen in the previous slide that the transporters normally have homo and hetero oligomerization. That is possible, right? So oligomerization in a different level. To have a finest transporter ability, it has to be most ordered. It has to have a most ordered structure. And we know that this 11 and 12 have highest similarity per se in its amino acid sequence. So which means that there is a possibility that instead of 11 becoming a transporter by per se becoming a homo oligomer, it can possibly become a hetero oligomer along with the 12, and then its function can change, which was actually the suspicion that we had to take up the further story further to know whether how this 11 and 12 in tandem sitting in the same cell is working to execute the function of whether to have more sucrose or less sucrose in the apoplast. So to prove that, what we have done is that we have taken the transporters from 11 and also 12, and we have performed the assays to make sure that at different sucrose levels, how this yeast growth is happening. So basically, whenever there is a possibility of homo oligomers, for example, 12 and 12, 11 and so this is a possibility of heteroligomers 12 and 12, and 11 and 12 in the other direction, and 11 and 11 in the something similar to that of a 12 and 12, homoligomerization versus heteroligomerization. You can see that whenever there is a heteroligomerization, you can see a much higher reduction in the yeast cell growth, which means these yeast cells were not able to take up and get the sucrose intake, which basically means that our original hypothesis that it is possible that 11 and 12 are interacting, and when there is a pathogen infection, one of the transporters, for example, this 12, in this case, what we are suspecting, is receiving the signal from a regular defense pathway, and that command is executed to form that, okay, you go ahead and form a heteroligomer with the 11 transporter, because if you leave the 11 transporter, it will anyway transport. And if you form, since of because of its higher similarity, this can form a heteroligomerization. So when there is a heteroligomerization, the satisfaction of biochemistry is done, that the oligomerization must happen for those proteins. So that happens, but the sucrose is not able to be transported. So this is a beautiful example that how these two transporters can work in tandem to make sure that to decide whether the substrate to be in which place, whether it should retain in the cytosol or it should get into the apoplast. So this assay was mainly proven to show that, and then later on we went ahead and proven the similar ones using the sucrose transporter activity assays. This using the radio tracer sucrose uptake assay, which is very similar to that one that I showed it to you in the previous slide. So you can see that the values, so you can just compare the y-axis. So here it goes like, hundreds, like up to 250. You can see that this is up to 120 and this is only up to 60. You can see that when there is 11 and 12, in whichever the orientation that it is, the Vmax is very less, and you can clearly see that the growth that I've shown you in the previous slides is clearly reflected in this. So again, this is to re-emphasize the point that it is possible that these two transporters can form a heteroligomerization to prevent the sucrose being transported into the apoplast. Now, the important thing that we are supposed to prove is that since most of this immune related stuff depends on some of the common regulators that are previously known, although we are now coming up with the story that a parallel immune response through restricting the sucrose availability or any other nutrition that pertains to the pathogen is possible, we still have to explain how a conventional immunity happens in the system. So you know that FLG22 which is recognized by FLS2 in the plant, these are the very well proven system that to explain the plant immunity, why a plant is resistant to a particular pathogen. So what we have done, we have taken the FLS2, which is a receptor, and how it is responding to FLG22 we studied. We simply taken FLG22 treated plants of these combinations, that is this 11, 12. So these are all, of course, labeled with either the N-terminal or C-terminal GFP fusions. And then we have taken non-inoculated and inoculated plants. The story that I'm trying to say is that whenever there is an inoculation, you can clearly see that there is an expression of this these transporters in the cell, which basically again means that, although I told you in the previous slide, I can re-emphasize again that this 12, which is a transporter that is actually regulating the 11, is possibly receiving the signal from FLS to FLG mediated plant immune system. I mean, that is the pathway. And then hence this is able to execute the function of final determination of where the sugar should demand, because the theory of sugar alone determined should depend on some of the clear signaling network, right? So one possible way is it is possible that this FLS FLG2 mediated pathway could be the determinant of final fate of the sucrose availability in the apoplast, which again determines the role of pathogens. Now I will just summarize this whole thing, and this story will not end. It is starting up with another important transporter called SUC2, but I will summarize this point in this slide. See, the first thing I would like to say is that I already explained to you how this sweet 11 and 12 transporters are localized. So it is localized to somewhere in the phloem parenchyma cells, to be precise, somewhere in the companion cell sieve element complex. Now, in the apoplast, if there is a less sucrose available, and this can really prevent the pathogen multiplication. And it is possible that this 11 and 12 can interact, either homo or heteroligomerization is possible to control this sugar levels, sucrose levels in the apoplast. Now, what I'm trying to say is that there is another transporter which I did not introduce so far in the talk called AT-SUC2, which is another key player. You can see that until phloem parenchyma, the 11 and 12, to some extent even 13 and 15, key players of putting the sucrose into the apoplast. So this is just leaving at that stage. If a plant restricts at this stage, the sucrose stays in the cytosol. For example, if this somehow the sucrose is already there in the apoplast, then the depletion is possible. It can also put back, but not these through transporters. There are other class of transporters called STPs which can put back, which normally doesn't occur. But the easiest way is through a regular phloem loading process. This phloem loading happens through another transporter called sucrose transporter 2, SUC2. So this SUC2 expression clearly takes away the sucrose from the apoplast and quickly puts it into the companion cell sieve element complex, which is loaded now into the phloem. Once it is loaded into the phloem, this bacteria that I am talking about today does not have access to the sucrose. So which is again a safer location. And more importantly, the phloem transport is the must for sucrose because it has to reach the zinc cells eventually. And that is the main purpose of the plant. Although I oriented the whole talk towards immunity, but for a plant, these transporters are normally used to perform regular sucrose transported into a destined cells of zinc cells to produce more seeds or to produce more root and stuff like that, right? So this SUC2 then comes the very important component of it. And it could also be possible that, as I told the story about this 11 and 12, whose regulation possibly restrict the availability of sugar entry into the apoplast, it is possible that the SUC2 also can tandemly work along with the 11 and 12 to quickly deplete. For example, this does not allow the sucrose to get into the apoplast. At the instant time, the SUC2 puts all the remaining sucrose into the phloem, which means that this apoplastic space is now cleared off with sucrose, which means that the pathogen has no way to go and eat its preferred source of carbon, right? So this is something possible, very good intercorrelation possible, which we are now exploring, that how these two SUC2 and the class of these two transporters can work in tandem to regulate deprivation of sugar in the apoplast. Now, as a first step, what we have done is we have just did a in situ hybridization to study the expression pattern of SUC2, which very clearly shows that its expression is again into the phloem region. And again, its expression is very clear that AT-SUC2 mutants did not have any sucrose accumulated, and we are able to clearly show that these mutants are able to grow less in the these mutants are able to grow more in the infected conditions, both with host and non-host pathogen. So again, I think I will probably not go much deep into the story and then finalize the whole thing by telling that there is a possibility that these two transporters can exist in homo-oligomers, or it is possible that if it is existing in homo-oligomers under the normal conditions, it transports the sugars, and the transported sugar is now exported into the phloem. This is the normal process. But if if it is possible that these transporters now are able to not able to do the function, then there is a disruption comes, which explains the whether the pathogen should be more or less under the infected conditions. Now, when you introduce the today's results, and you see that the red one is sweet 11 and the yellow one is sweet 12, it is now, instead of homodimer, it can also form heteroligomer. So this heteroligomerized proteins now can reduce the availability of sucrose in the apoplast, and hence we are able to protect the cell from the pathogen infection. Now with this I will stop, and these are the team members that that are present in the lab. But of course, as I said, the work was entirely done by Uruj Fatima. And I would like to now answer any questions, probably anything ranging from very generic to all the way to the specific questions about the transporters. But I would love to get responses from students, any basic points that you would like to see. So if you want to summarize, I can also make one more time a brief summary of what I said. If it is really inundated with a lot of data on your mind, that if you are not able to perceive it, I can do it again. Let me know the questions, if any. Thank you. Yeah, Gitanjali, go ahead. Yeah.
Actually, you were told about there is actually resistance. Resistant, right?
If the absence of, if the plant does not have a leaven, yes. Yes. So actually, this is a relative comparison. When I say resistance, so the apoplast is never full depleted of sucrose. The constant exchange of sucrose happens in a regular continuum by the cell, and it need not be only with 11 and 12. It can be with 13. It is also possible with 15, which are also doing the very similar transport. But leaving that point out, just for our own from our own experimental data, we can explain like this. For example, if there is already a pathogen oncoming expected in a wild type plant, the scenario of how these two transporters will function when they are together in a wild type form, like no disruption, may be completely different when they are working, especially when tandem function is existed. However, when you make one absent, maybe the other is completely behaving in a different way, which we are still not able to explain why it is behaving. But there is a systematic possibility that these two transporters are still in, I mean, it is feeling that the other guy is not there with me. Now, what should I do? It is trying to do. Maybe it is overdoing it, or maybe it's trying to settle down. So we have to find out what exactly during that transit part of time it happens. But leaving that small part of the story unanswered, the remaining larger question is clear. Less and more multiplication in terms of wild comparison to wild type is clear that whenever we have absence of 11, it is not supporting the bacterial multiplication. Whenever there is an absence of 12, it is supporting the multiplication. So if I retune my word, I can stay like that. But it is possible. There is something going on in that which we have to still explore it. And we need more of a complicated taglines. So more than your question, actually, we landed up getting into the other two transporters like the 13 and 15, which I never introduced, although in the very first slide I showed it, but afterwards I never carried over that point. So those two still remains, which probably is the bigger players. But now since the story is proceeding further, the SUC2 is actually the player that it is now we are trying to deal with. So there are two other outstanding questions apart from yours that we still have to address to make sense out of what exactly goes on in this region and who is actually deciding to load and how much to load into this. Yeah. Yes. So far, actually, we are thinking that 11 is a master switch, but it looks maybe it not. Actually, the reviewers are equally arguing that why not 11 is a master switch. And it's not necessarily the 11, but maybe who's controlling these two. Maybe they are the ones that maybe if it is FLS mediated signaling, then it could be that. So it goes like that. Yeah. Since it's a dynamic environment, the apoplast regulation, I have not introduced the STPs, sugar transporters, which puts back some of the sugars back into the cytosol regularly. So I have not introduced that into our scenario. And apart from that, there are other candidates that I just now told you. So it's like really a very complex place to deal with. But overall, we are able to show that sugar deprivation reduces the bacterial multiplication. Thereby, it is able to bring resistance to the plant. How? Yes, there are a lot of, because these questions are remain to be answered and must be answered because there is no particular way to regulate this sugar flux in a bigger way because these are all regular metabolism. If you disturb a regular metabolism, then the plant will not like it. So then needs this question like this to be answered to find out a real tuner and whose regulation will only tune it, not disrupt it. Yeah, others? Any other general questions? Yes, Kumar. Very good question. Actually, just when our work was going on, that is one of the reasons that my student also quickly went to postdoc. There was one Nature Plant paper that shows in the root Why? So we all know that the drought condition, the root shoot ratio is completely altered, and it towards the root, like root biomass always increases under the drought conditions. Now they have come up with a proof that the transporters both of the transporters that I am explaining are actually the reason for explaining the discrepancy in the root shoot ratio. In other words, higher root biomass. Now the story is completely, now what we were going in was all with aerial parts, like something thinking about the oncoming photosynthetes and its translocation and stuff like that. Suddenly there is a story that is now entering that how these photosynthetes are regulated at the root level, which is a very interesting question. And they have just run short of discussing or adding data on the root pathogens. But I guess they are already working on it. But we know that when we tested with the root pathogens, the story is completely different. And so now I have to really think how these transporters in the root, which is non-photosynthetic and mostly zinc cells, now are going to work on it. And how this dynamic response of sugar limitation, whether it will fit as like a photosynthetic organism in the roots also. It's possible. I mean, it is sensible to expect that the roots also can have a similar strategy, but I don't know, because the dynamics when there is a photosynthesis going on, the uptake and the transport of sucrose and its flow is whose dynamism is different. However, in the non-photosynthetic systems like root, whose depend on these photosynthetic cells to receive sucrose and whose influx is completely restricted or decided, how whose dynamism, how it will be dynamically responding, I don't know. But definitely it is a very important question, and it's also interesting to know that. And this paper is actually gave away a lot of our data in terms of the protein story and its interaction and stuff like that. Although I presented it more keeping the pathogen always as a main backbone and presented the story, so it's flowing fine. But this story, when you take it apart, let's say take out the pathogen from the story, then most of the stuff that what they have demonstrated is in a different way. They have demonstrated the similar things for these two transporters in terms of its function, but in the root. Yeah, any generic questions in terms of its application in disease control or from students?
Is it possible to make the way for epiphytes enter into the plant as endophytes through these transporters?
Yeah, so that's again another interesting question, but I don't have an answer from an epiphyte, but I have it from rhizobium. So basically, since the rhizobium is an endosymbiont and there are clearly worked out signals of how this rhizobium enters and it becomes, there is a clear cooperation from the plant. So in that, in Medicago, they have shown that sweet leaven is the key player in determining how much sucrose to give it to rhizobium and how to give, when to give, and how to keep it as a symbiont. Now, if I extend that to explain it for an organism that is coming outside and staying as an epiphyte, and then you are expecting that if it's a good organism, if it becomes an endosymbiont.
Or for example, when you are staying in endophyte, how they enter inside the plant, now we are telling only about it is very restricted one. Sometimes the size also varies. Through the transporters, if it goes, so is it possible to target all the cells?
So the organisms doesn't go through these transporters, but it is actually completely either restricted or attracted by the action of these transporters. It is possible that these transporters can dictate in this particular location whether the microbe can stay or not. So, for example, if you want to extend your question that whether the epiphyte becoming endophyte and if it becomes a good supporting point, let's say we are artificially applying it and you want to make sure that it stays in the plant and so that it provides durable, benefits to the intention of application. Now, if the selective induction of these transporters are possible in a place where you want to have your endophyte to do its job, it is possible. So if you understand thoroughly what are the transporters that are responding to this endophyte, and maybe a transient way of doing it for a certain duration of time until the beneficial function to be reaped. So after reaping of the function, then the whole thing can be shut off and then used it for regular metabolism. It is possible. The transporters can be a driving point or dispersal point for deciding the location of or at least the quantity of that particular microbe in that location. It is possible.
If you apply in soil, is it possible to take it in the phloem layer alone? Phloem specific proteins, is it possible? Localized
So you want to have the endophyte in the phloem.
phloem.
You want to have it in the phloem. Okay. There are sweets that are specific to xylem. I'm not sure about these are all loaders, but they are not actually.
Not sweet. Anything through this transporter, it is phloem specifically express that protein. So that phloem feeding insects like white fly, aphid, or any kind of thing, narratives that do some mechanism.
Okay.
I have to see that aphid and narratives mechanism. They are expressing it only in phloem.
And you want to, let's say if it is completely depend on phloem, which indeed depend on these nutrition source from the phloem, and you want to short term regulate that and make sure that that doesn't like that leaf anymore, it goes away from something. It's possible. Like maybe we can feed these Arabidopsis plants because these are all infected heavily by aphids and see whether they show any difference in terms of it is possible. Yeah, it's a good idea. Students, at least, maybe I'm going to lock this room. You have to ask a question. Yes, please. Yeah. Yes. Yes, yeah. Let me publish this paper, then I'll come to SUC2. I will say that SUC2 is important. But anyway, on a serious note, your point is well valid. It is highly possible transporters like SUC2 is more efficient than these two transporters in terms of taking away the sucrose at the site of infection. It is possible. And since the SUC family has 8 members, of which SUC2 is the potential phloem loader. So the reason that plant did not have more phloem loaders, unlike the sweet families, it kept other candidates in place in this location, but in SUC family did not keep. So there is a specific reason that if this is the case, it is possible that maybe that is the strongest transporter that can do the job. So instead of manipulating sucrose, sweet, one could work with SUC2 as well. But your point is well taken. Yeah, you're right. So ultimately, that can be a potential downloader of sucrose into the phloem. Very heavy roles. Actually, I've shown only a part of data. As I shown this, whenever I explain this thing to Gitanjali that there is a dynamism, there is also dynamism in terms of conversion of one sugar to other. Sucrose doesn't remain sucrose all the time in the upper plast. And these invertases are the heavy players of converting one form to other. And the second point is that these invertases are the determinant of another class of transporters called STPs. The moment these invertases start acting, there is a signal that the STPs gets induced and they are actually involved in putting back sugars in a different places, maybe back into the cytosol, something like that. And the third thing is these invertases are also heavy inducers of sugar sensing and sugar sensing mediated immune responses in the plant. So these invertase actions are actually heavily monitored by the plant to decide on many features. So they could well be a player. As I explained that it could activate STPs, it could activate general sugar sensing and sugar level mediated responses in the plant, which could be part of our immune responses, which are all unexplained in all these mutants. By the way, that could be a very good study to go. So if you recollect, I never showed anything about STPs. I never showed about invertases. I just started introducing the SUC2. So there are more players. Definitely inverters are a important components, I mean, players to look at in this story itself. Yeah. I succeeded in making one student ask question. Then the rest, I think you can decide. Yeah.
There are recent theories for virus elimination to apply more micronutrient in the soil. The plant is able to withstand, and the hypersensitive response, we knew that. But besides that, is there any mechanism to regulate the viral multiplication?
Yeah. So actually, this is completely a different question. So I will not connect this to the sucrose or any nutrition story, because we know that viruses doesn't come for plant for nutrition. It just comes for multiplication. It doesn't eat any carbon source from the plant. So this can be a totally different question. So there are mechanisms that, for example, if a virus is specifically acclimated to phloem, and then the phloem connected other parts, doesn't actually necessarily get into a lot of mesophyll cells, lot of other things. So when such a restricted carve of from the delivery, for example, if an aphid delivers into the phloem, and then thereafter wherever the of course there is a connection to sucrose here, because in terms of wherever sucrose goes, this virus also will go. So but the main point here is that such restriction can be exploited to answer your questions. Maybe why clearly an anatomical distinction to make the virus restricted to some places can also be exploited. But unfortunately, these viruses are now I mean, shooting the plasmodesmatal limits. So we know now that the restriction was largely possible by the plasmodesmatal size exclusion and a few other aspects. Now it started even after coming through the phloem. Once it enters into a cell, then cell connections through plasmodesmata is always there. So now it started entering into other cells also. So yes, you know that the virus is much evolved faster than any other eukaryotic system. So yeah, so I can only think of something that is very similar to the anatomical features, that how this plant is able to restrict them against a particular class of virus can be extended to, let's say, CMV, or transmitted by not by whitefly, not by aphids, which are all transmitted. So it is possible. Yeah, I'm thinking. So no, I got your question. So the point is, why in the first place heterooligomerization? And that I explained it, that it is possible that one wants to regulate the other, and it wants to decide, despite meeting all the oligomerization requirements of a protein, you still want to control the sucrose, and hence you wanted to have a heterooligomerization. Now your question is that how this heterooligomerization is determined, and is it because of protein levels? And yes, one obvious expected answer could be the levels, because now it can titrate out depending on how much of the 11 is produced and how much of the 12 is produced. Somehow there is a sensing that, okay, whether now I should go for homooligomerization or the spillover should get into the hetero. The very reason that the biology is producing spillover means it has to be used somewhere and for some other purpose. So that purpose could be a heterooligomerization. So that is one possible answer. The second possibility is that whenever there is a heteroligomers formed, we know that there is a less efficiency in the system. This is one of the reviewers, tightest arguments, that, so why a plant first should form a heteroligomer? Because once it forms, it also, even after pathogen, it will stay for some time. And maybe this is that if it is a by default function of these two transporters under normal conditions, if there is, it's not that only the pathogen is altering the levels of sweet 11 and 2. Maybe it's also responding to other things. So in that case, also the levels will vary. And in that case, also heteroligomerization will happen. But then in those cases, the sucrose limitation was not necessary because there was no pathogen. So this is actually a little complicated thing if we have to explain it through only the levels. So it is possible that these ones can have some kind of a post-translational modification, which could, I don't know, it could be sumylation, it could be acetylation, it could be phosphorylation. I don't know, something like that is being somewhere in between acting, which is actually so I can still think about phosphorylation, because in that Nature Plant paper which I showed for the same transporters, where the SNRK is the one that they actually use, phosphorylation decides how much of the sucrose goes into root. So there is possibility that one of these post-translational modifications are playing the role. Whether one of these, the strong contenders in this are in the rank of first is phosphorylation, second is sumylation, and then there is a possibility of acetylation also. One of this could be a determinant of this, whether there should be a heteroligomerization or homoligomerization. This is all hypothesis, but it's a very good question. It's actually a physiology topic. Phloem transport. Thank you all. Okay. Okay. Actually, there is a question from online, it seems. So, how do I read it? In that level. Okay. Do SUC2 expression is high during infection or same as normal? It is high. So, again, the SUC2 expression we have actually selectively did with in situ. In one of the slides, when I quickly passed through it, I showed it. So the reason why we went for in situ hybridization instead of a regular thing is that it is high, but maybe it's not the overall tissue status of high that will explain the question. So that's why we went for a in situ specific thing, where in that specific location where it is acting, it is explainable. So the answer to that question is yes, it is high, and it has a small degree of variation between the response to non-host pathogen and the host pathogen. So that is one of the reasons that why we thought that maybe during the pathogen infection, the plant is trying to upregulate the sweet to get the sugar out of apoplast. So yes, it is high. One question. Okay, thank you.
On behalf of CPMB, we are much thankful to Dr. Sendil Kumar for his excellent lecture. So we are presenting a small note on behalf. So it's a time for formal vote of thanks. So on behalf of CPMB and on my own behalf, I am much thankful to our Vice Chancellor Madam for granting us permission to conduct this guest lecture. And also we are much thankful to the guest speaker, Dr. Sendil Kumar Muthappa. He is all the way from New Delhi, and for his excellent lecture. And I think you all know, like from the introduction of Director CPMB as well as Dean SPGS, he is our own alumni, did his BSc in agriculture, and then he moved to US Bangalore for his under ICR fellowship, and then for PhD with Oklahoma State University, and again he has back to India and working in NIPGR, in a reputed national institution. So we are much thankful to Dr. Sendil Kumar for his lecture, as well as motivating the students for future collaborations, as well as for the postdoctoral, as well as other PhD fellowships. Thank you, Dr. Sendil. And also, I'm much thankful to our Director CPMB as well as Dean School of Postgraduate Studies for his constant motivation and encouragement to conduct such kind of guest lectures for the benefit of students, as well as for his contribution in terms of finance, as well as providing us this hall facility. My sincere thanks to our coordinator, Dr. Raghu, as well as Dr. Kritika for coordinating this conduct of this guest lecture. I'm much more thankful to our senior faculty members, Dr. Sudhaga sir, Dr. Mohan Kumar sir, Dr. Ramalingam, Dr. Arul, and other CPMB faculty members for their presence. And also I'm thankful to Dr. Uma Madam, Professor and Head Biochemistry, for her presence. And I'm much more thankful to the Plant Physiology Department, Professor and Head, and the students for their timely presence for the guest lecture. And also the Professor and Head of Department of Plant Breeding and Genetics for sending their students, as well as the plant pathology students for their patience in attending this guest lecture. Once again, I am much more thankful to all the members of CPMB, as well as the others who are directly or indirectly involved in conducting this guest lecture a more successful. Thank you once again. Thank you all. I think the refreshments are arranged outside the hall.